Hello and welcome to Agri History. Today we will cover the topic of chickpeas. Caesar eritum, also known as a chickpea, is a member of the legume, pea, or pulse family, which includes beans, peas, peanuts, and many other plants. Chickpea is considered the third most important legume crop in the world, and today we will cover the domestication of chickpeas. Chickpeas were domesticated alongside many other crops such as wheat, barley, rye, peas, lentil, flax, and vetch in the Fertile Crescent between 10,000 to 12,000 years ago. Initially within this region, ancient hunter-gatherer tribes would harvest wild plants for food, but these groups would eventually move into agriculture, with the earliest sign of agriculture being 7500 BC, and earlier dates being plausible. One of the earliest records of chickpea cultivation is in Kayano, Turkey, with samples that date between 7500 BC and 6800 BC. These would be likely wild chickpeas rather than domesticated strains. Its use as a cultivated crop, however, is well documented from 3300 BC onwards in Egypt. The progenitor of this species would be Caesar reticulatum. This species is found only in southeastern Turkey, where domestication likely occurred. In that area, the two species are interfertile, and both the domesticated species and the wild species have similar seed proteins and morphological features, although some versions of the domesticated form differ in random mutations. In the secondary gene pool, there exists only one species of Caesar. This species is called Caesar echinospermum. This species differs only by one mutation called reciprocal translocation. This is where a portion of one chromosome swaps places with another portion of another chromosome in the same genome. This species can hybridize with our domesticated species of chickpea, but will often produce sterile hybrids. Chickpeas are mentioned as both a food and medical plant by Homer in the Iliad, which was written between 1000 BC and 800 BC, as well as various herbal literature in ancient Rome, India, and medieval Europe. This crop spread with a cluster of other founder crops that were mentioned prior from the Fertile Crescent into Europe, as well as Central Asia and onwards. This began around 5500 BC. Chickpeas appeared in India either around 2000 BC or the first millennium BC, depending on source. The changes that came from domestication were the loss of dormancy, reduced pod shattering, larger seed size, larger plant size, and variants with more erect habit, as well as reduced pigmentation within the beans themselves. The biggest change was a change from a winter habit to a spring habit. This resulted in a reduced pathogen attack on the crop. The domesticated chickpea can be divided into two groups: the large-seeded Kabuli group and the smaller-seeded Desi group. Based on current genetic evidence, the Kabuli probably evolved from the Desi type. The Kabuli type have large pods, large seeds, large leaves, and taller plants, but they also have a narrower genetic pool to draw from. The Desi type has a large genetic diversity, but also small pods, seeds, leaflets, and small stature. And they have a wide range of flower colors, pod colors, and seed colors, as well as variation within seed and surface shape, while Kabuli types have a primarily white flower phenotype, with other colors showing up very rarely. The Kabuli type is restricted to the Western Mediterranean in range, while the Desi type is found in the range of the Eastern Mediterranean to Central Asia and the Indian subcontinent. The primary center of diversity for chickpea is the Fertile Crescent, where this crop was domesticated. With the spread of this species, secondary centers of diversity have shown up. One formed 2,000 years ago in Mediterranean Europe, and another in the same time frame showed up in the Indian subcontinent, and a third, again in that same time frame, showed up in Northeast Africa. New centers of diversity have also shown up in Mexico and Chile ever since Columbus introduced chickpeas to that area. Based on current genetic data, the greatest level of diversity in chickpeas is found in the land races of India, followed by the land races of Iran, Ethiopia, Afghanistan, Pakistan, Turkey, Mexico, Syria, and Chile, with the location with the least amount of genetic diversity being the Soviet Union. Due to the land races having a patchwork distribution in those areas, and the limited introgression of wild DNA into its domesticated form, and the switch from winter growth habit to spring growth habit, seem to indicate that chickpeas have evolved from a very narrow genetic range, although with a wide selection pressure for different ecological niches. This concludes our video on chickpea domestication. Stay tuned for our next episode on dry bean domestication. Ciao!